Thank you very much, Sarah. So I will ask the other speakers to go ahead and unmute and turn on your cameras. Great. There were a whole bunch of great questions, and we'll try to summarize a few so that we can get to some of the key points. So I think maybe starting with some of the trends and topics that Carlos brought up in your talk around endothelial cells. And maybe, Carlos, can you comment a little bit more on the expression of ACE2 and perhaps other entry receptors in the endothelial cells versus the pericytes? And then as an extension, do you see any evidence for actual SARS-CoV-2 infection or presence in those cells versus just expression of the entry factors?
Yes, so we have seen by electron microscopy and now also with immunogold electron microscopy using antibodies to the spike and the N protein, the presence of viral particles in endothelial cells, but not in pericytes so far. So I think that maybe the amount of viruses that can be seen using these technologies, it's too low in some cells, but definitely we know that endothelial cells have viral particles, and we also have done it by in situ hybridization and immunohistochemistry in the same tissues.
And then Sarah, I don't know if you could maybe comment on whether there's been efforts in some of this meta-analysis and clearly looking at the ciliated cells, but maybe looking at some of the other cell types and lineages, including the endothelium, of what this looks like across different tissues.
So in terms of the healthy reference, I have to say that ACE2 doesn't seem particularly high in endothelial cells themselves. I mentioned pericytes, and I wonder whether maybe there is infection by that cell population. For the post-mortem tissue analysis, I think it's a little early to say, but I know that there are studies underway to delve into that in more detail. In the UK, Omar Bayraktar and Sanger is doing work on that, and presumably also in the US and elsewhere.
Great. So I think another area that a good number of participants were curious about is, looking at this data both in terms of thrombosis and the effects in the endothelium elsewhere, but also, from many reports, relatively low levels of the virus or entry receptors in the blood. And so maybe just to go around the panel a little bit and discuss, how you're thinking about the transmission of the virus to various organs, and whether there's a role for the endothelial cells or maybe additional cells or questions that really remain to be clarified.
So if I can start, I can say that definitely there are not many ways from a virus that starts in the nose to go into the kidney. So the highways are very clear. So we know that viremia occurs. We know that not all of the endothelial cells, like not all of the epithelial cells, are similar. And as we said, from anatomical, a structural point of view, there are some blood vessels, essentially capillary vessels, that they are non-fenestrated, they have very tight junctions, they have a very specific profile, including some detoxification proteins such as the transporters, P-glycoprotein, etc., and ACE2 receptor is part of this phenotype. So in the same way that we have different lymphocytes, that we have different epithelial cells, we have different endothelial cells, and this is something that these specific studies at comprehensive look at endothelial profiles in different organs and single cell analysis are going to be very critical.
So if I could chime in there, I would, sorry. If I could chime in, I would say that in animal models, it's clear that there is no natural tropism of the virus outside of the respiratory tract. It's very, very confined to the upper respiratory and lower respiratory tract. However, even in our own sequencing data or attempts to do plaque assay, you do find, sporadic hamsters or a sporadic ferret that tests positive in a heart or in a liver or in a kidney or in the brain, which I do think very much mimics the heterogeneity of the diseases we see in reality. And so I would argue that what's probably happening is that once the damage to the pneumocytes is done, there must be barrier breakage that will allow the virus to get into the bloodstream, but that is not common. It's not part of the natural biology of the virus. And for that reason, like, should that happen, I think the vast majority of it gets neutralized or goes nowhere because ACE2 expression is not everywhere, it's not ubiquitous. But when it does land on a tissue that it can actually get into, that's when we see these really unusual cases emerge. And just as a case in point, we work with a lot of human organoids and gene-on-chip models here as well. And if you take a gene on a chip, like a heart gene on chip or a heart organoid, and add virus to it, it will like completely liquefy that culture. So clearly the ACE2 and cardiomyocytes, as an example, is a cell type that this virus loves and can replicate and destroy, but we don't see a lot of, there's 2 reports published about identification of virus in the heart of patients. We've checked with Cornell lots of autopsies, and seeing actual virus in the heart is really, really rare. So there's an example of, if the virus is in fact circulating in the blood, it's stochastic whether or not it hits on a tissue that it can actually establish an infection in.
Can I just add that, like, we don't see ACE2 on any of the blood cell types. Could it be macrophages that are like eating an infected cell or something like that?
I would say that's a dead end. So I think it's literally the virions in the blood, which is definitely not a very efficient way to get into any kind of tissue.
It's just virions. Yeah. Okay.
Maybe, Sarah, while we're here and thinking about the progression of the disease, I wonder, if you think that what others have said about this topic kind of in the adults is, likely true, or if there's this kind of extreme of the Misty patients that might add to the complexity or maybe provide some insight into how the spread and infection is progressing.
Yeah, it's a great question. I think Misty is a really interesting case study. It's something that I know scares a lot of parents, but is incredibly rare. And so from my perspective, as someone who focuses on pediatric immune deficiency, I kind of come to the point of, as I mentioned, if something is incredibly rare that happens in the context of infection, wondering how different the host is. Well, there could definitely be pathogen components. It seems very likely that we'll find differences in the kids, whether they be genetic or in sort of the baseline immune regulatory state they may have that may not be genetic in origin. What's fascinating is that the most clear definition of Misty patients is that they're healthy at baseline, that these kids are generally not with any other chronic medical issues, compared to our COVID-19 patients, especially our severe COVID-19 patients, tend to have other chronic medical issues that are ongoing. They are rarely perfectly healthy. So it's this really fascinating situation, and incredibly intense disease that comes on acutely but late in the course of an infection, where we may be able to learn a lot. But it has been challenging to get some of these mechanistic questions in the same way that is being done in adults, just because there's so few of these children to really get at these. So I think there's a lot left to learn, and obviously the animal models are not focusing on the pediatric component either. So I think there's a lot to do. And I think there's a lot of really interesting data out there that we can start getting closer to mechanism, but there's so many open questions that are left to be investigated.
So there was an additional question that came in, curious about the sequencing of events. So, some of the talks mentioned that acute respiratory distress syndrome appears to be preceding the spike in cytokine. And while many have focused on the cytokines themselves, whether it could be that maybe the cytokines are indeed not the real problem. So could maybe, again, starting with Sarah, thinking about Misty and the interaction of the immune system, thinking about that ordering or kind of open questions as you see it, and kind of understanding how the sequence of event and the pathology is playing out.
Yeah, it's a great question. I think, all of us would love to have samples from these patients before they got sick. That's one of the main issues we have, right, especially on the human side, is that we don't know what the baseline was. One of the things we've been talking about on our team is that the other issues, that the reality of our samples is that probably at least 36 hours into when someone is admitted and has symptoms, whether it's COVID-19 or Misty. So trying to get as early a sample as we can is another focus, because we know in Misty we're missing likely weeks of the interaction between the immune system and the virus. However, what we've seen in an earlier paper was that in Misty specifically, we saw more of a role for IL-10 and TNF in our patients as being key. And because of the nature of the samples, we don't really have as much of a sense of the timeline. Like I said, what's interesting between the COVID pediatric and the COVID Misty is that the immune dysregulation that we see sort of throughout the illness in adults sticks in our pediatric COVID-19 patients. But for Misty, while they have a really significant hyperactivation in their CD8 T cells and CD4 T cells, it tones down as they come back to a more normal clinical state as well. Again, maybe reflecting the fact that they were healthy at baseline and may not have as much dysregulation at baseline. But we really need to get to those recovered samples and get more in detail in the transcriptional pathways in these cells to get a better sense of what's going on. So I think I have less to say. I apologize about the timing.
And Carlos, could you maybe comment on that? I think that your talk certainly.
Sure. So like in cancer, some patients may present already in a stage 3. So the type of a staging and model that we are proposing, even though it looks linear, everything has its own plasticity. But in general, yes, we see that the inflammation comes a little bit later, and it's not a single cytokine or interleukins. It's for the reason that maybe addressing with a single agent to for example, block IL-6 may not help, because there are other also parts of the pathways that they are very much activated. So it can make patients very sick. We have seen that the use of a specific blockers may help, but we need much more of an approach that is going to be more pan-blocking than just a specific cell types and of interleukin types. And of course, as that is progressing, it also produces a stress in the endothelial cells. It's well known that cytokine storms produce endothelitis themselves because of all of the receptors that the endothelial cells have against some of these critical inflammatory components.
Great. So maybe one last area of discussion for the group is around models. So maybe thinking about the complexity, of course, and understanding the progression of the disease in human patients, but the promise of utilizing different model systems. So Ben, you discussed the hamster and also some organoid work. Would be curious to hear a little bit more about how well you think that they model the disease, and also maybe if there are specific gaps that you're seeing and trying to work towards.
Yeah, no, sure, happy to. I would say that models are exactly that. They all have limitations. The mouse model requires modification of the mouse and modification of the virus. The ferret model seems to be restricted only to the upper respiratory tract, which is not something we see anywhere else. I do like the hamster model the best with regards to its capacity to be systemic and, to a lot of the questions being posed, like when we're talking about looking at people, there is no way of seeing what baseline was. And so, animal models are important in that we have the capacity to at least control those variables, even though obviously they're imperfect. There have been a number of reports that as hamsters get to advanced age that they also develop COVID and it becomes a lethal infection. The problem is that getting old hamsters is tricky. An advanced age in hamster is about 2 years, and commercially available hamsters are 4 weeks old. So we are aging hamsters to see if that will improve. I have to say that organoids and cells have proven to be a great first line of determining how the biology of the virus works, maybe testing some drugs for down selecting which ones might have therapeutic value. But I sit on a very large DARPA consortium where we get handed a very short list of 20 to 30 drugs that have gone through multiple high throughput screens from places all over the country, even the world, that look fantastic in organoids, they look fantastic in cells. We determine the PK value, the biodistribution in hamsters, we put them in and they do nothing. Hydroxychloroquine is the best example of this. That's something that works beautifully in cell culture, but just has no efficacy in vivo because it just doesn't get to the right spot or doesn't get to the right concentrations. And so, having an animal model is really important in our capacity to test drugs and therapeutics and prophylactics. And I don't think any cell system, we're not quite there yet. I don't think organ on chip or organoids really get us to that point because they really do provide us a lot of false leads.
Any others want to comment on the utility of models? Or, Sarah, I know that folks within the Human Cell Atlas have also done some comparative analysis of, the Bena profiling and at single cell.
Yeah. So, I mentioned, in my talk just now, I mentioned mouse human and for the oral cavity, and there's a lot of conservation, across it. And then obviously there's macaque, so rhesus macaque, and there's been work from many different countries on that. And in the Human Cell Atlas community, the the profiling of the tissues from the rhesus macaque, you know, for comparison with human. And I think that's obviously a difficult one, but it seems like a really good model. Yeah.
Great. So with that, I think we are at time. So again, want to thank all of the speakers and wrap up this session. We'll take I think a 5-minute break and come back at just after 12:35 Eastern time for the next session. So thank you again, and enjoy the rest of the symposium.
Great, thank you.
Thank you.
Thank you, Donna.